COVID-19 is a global pandemic that's killed over 375,000 people around the world, and we desperately need treatments for it, but we don't have the 5 to 10 years that it normally takes to develop new drugs and get them into clinical trials. So the fastest way to develop drugs to fight COVID-19 is to repurpose existing drugs that are already on the market or in clinical trials. And the big advantage of this approach is that you can leverage a lot of the research that's already been done on these. So these compounds have already undergone extensive testing in humans, so we know a lot about their safety. And that means that if we have a good reason to believe that they're effective, we can advance them into clinical trials almost immediately. So to manage expectations a little bit, we are not expecting to find a cure for COVID-19 sitting in someone's formulary. What we're looking for here are drugs that are good enough to buy us time until something more effective like targeted antibodies or a vaccine comes around. So the first thing that we did was we mined the literature for therapeutic targets. And the goal of this was to develop a working hypothesis. What protein are we going to target? Why do we think it's going to be effective? And what evidence is there that this is actually going to work? We did this using a tool called Dox2Graph that we developed in our lab, which uses natural language processing to generate a map of relationships between chemicals, diseases, and genes discussed in a corpus of documents. So this map generated by Dox2Graph also works as a content indexing system that lets you retrieve sentences and documents of interest. Basically, we get a bunch of sentences that tell us about this protein, right? And we saw that TMPRSS2 looked like a very promising target because it affected multiple coronaviruses and seemed to be necessary for cell invasion. So now we have this mechanism of action that looks very promising, right? To invade cells, TMPRSS2 needs to cleave and activate the coronavirus spike protein when it's binding the ACE2 receptor, and that lets it get into the cell. Okay, so what we want to do is we want to disrupt this process by inhibiting the activation step. And what we want to do now is find existing drugs that can bind and inhibit our target, in this case, TMPRSS2. So here are the top 6 repurposing compounds. 3 of these are currently marketed, meaning they've been approved for use in humans and they're being prescribed to patients for other conditions. The other 3 are not approved, but have made it at least to phase 2 clinical trials. So they're not, on the market or available for prescribing, but they've already been cleared for testing in humans in clinical trials to test effectiveness. Now, one thing I would like to make very clear, these are preliminary results when you get them out of computational simulations. In the hierarchy of evidence, computational predictions are pretty low. So you can use them to figure out what experiments you should do in cells or mice, but it would be irresponsible to test in humans without experimental validation. So that being said, we're very excited about our results, and we're currently testing our candidates in cell models and direct binding assays and laying the groundwork for clinical trials of successful candidates. Another exciting development is that we're working with a number of students in the CS448P class to scale up some of these simulations to go through a larger library of compounds and pick up anything we missed and generate additional additional lines of evidence, as well as look at additional targets. Yeah, so we've had to, do a lot of, networking with other labs to, find collaborators. And, it's non-trivial to find people who can express and test the protein in direct binding assays. And also, to find BSL-3 facilities who can do the pseudotype virus assays. And if we wanted to go into, live virus at BSL-4 facility, getting into those is not easy. And, in terms of the incentives, right? I mean, it's almost like, to me, it feels like we're really the ones who have the incentive and who need to drive it. So on our part, we've been very active in trying to find collaborators. And for clinical trials, it's, similar thing where there's lots of clinical trials for lots of drugs going on. So you really have to kind of champion or find somebody who wants to champion the development and the movement of this, into the clinic, into the front lines.